Excellent. So sharing my screen at the moment. So what I'm going to do is a little bit of an introduction to the way that we're going to run the webinar this afternoon or this evening for those of you in New Zealand. So I'd like to thank everybody for joining in, and particularly for those in New Zealand, because I know it is later. So we're probably cutting into dinner time. So this information that we're going to deliver today is from a specific project that we've been running, and there's a reasonably large project team and a very strong project team, I would say. You've got the names on the screen there. My name is Sharon Taylor. I work for an organisation called Plant Health Australia, and we're here today to share information on a project that we've been running for improving preparedness for a pest that's exotic to Australia and New Zealand called spotted wing drosophila. We've got quite a big team, as I've said. It's a collaborative project between Plant Health Australia, Plant and Food Research, which we'll alternately call Plant and Food Research New Zealand, and it's also got an Australian arm, and Cesar. We've got in-kind support from Hort New Zealand and members of a steering committee. As I have mentioned, it's a project funded by Hort Innovation here in Australia. We've got some excellent collaboration. In today's webinar, there's a few things I'll do as housekeeping. So we're here to share knowledge and information on this project, really to support our preparedness for response and management to this exotic pest, which has emerged worldwide as a very significant issue of soft skin fruit. Just for the purposes of the webinar today, because we've got a large number of people online, we've actually got everybody on mute except for those who are talking. There will be opportunities to ask questions, and we'd like you to do that. Hopefully you're familiar with Zoom for the chat function, which, if you can see it on screen, if you're using a computer, if you hover over the screen, there'll be a taskbar at the bottom, hopefully, which will show you a chat function. You can put your questions to all panelists and attendees, or just to individual panelists if you prefer. We're going to run tight on time, so we'll try and answer questions using that chat as we go. And if we run out of time to answer all the questions, we'll actually be posting up questions, and we can respond to them after the webinar. So what we'd like to do to kick off and get started, we want to try and benchmark, I guess, where we're sitting at the moment. So we're going to try a bit of technology in this webinar. We would like everybody to rate their knowledge, or your knowledge, current knowledge, on spotted wing drosophila. The response options you've got are there in front of you. Hopefully you can click against whichever one most suits you, either extremely low, moderate, high, or extremely high. We understand that some of you will have extremely good knowledge of spotted wing drosophila, and hopefully some of that is from information that we've already been sharing in this project. So I hope the technology works. I'm going to move to the next slide because we are going to run on time. And so what I'm going to do now is, while that poll is still running, I'm just going to introduce the speakers for the day who are part of our project team. So we're going to kick off with Dr. James Mayno, who's a senior research scientist at Cesar, and he's going to talk about establishment, spread and impact of spotted wing. We're going to move to another one of our team members, Dr. Lloyd Stringer, who's going to talk about surveillance, and then I'll be back again to talk about management options and what we've learned from overseas in their experiences managing spotted wing. So moving on to hopefully, excellent, the technology is a success. So Jessica lies running this in the background, so thanks to her. What we've seen so far is that most people feel that their knowledge of spotted wing is low. We're hoping during the course of this webinar and through other material that we're going to circulate from this project that we will be able to improve on that in terms of knowledge. So thank you very much for actually participating in that poll. And now what I'm going to do is hand over to James, who's going to share his screen, and I'll put myself on mute.
There we go. Thanks for that, Sharon. So as previously mentioned, today I'm going to present some of the work we've been doing on the establishment, spread and impact potential of spotted wing Drosophila, with a focus on Australia. So I suppose I'm going to break up this talk into 3 distinct sections. So we're going to have the establishment and spread and impact separately as 3 points. And I think the reason for that is because just because there are conditions that support establishment doesn't necessarily mean that the pest will occur there. It needs to spread there. Just because something can spread there doesn't necessarily mean that there will be impacts caused. So we're going to look at each one of these things and build on them as the talk goes through. So starting with establishment potential. Well, I think what is plain to see with this pest is that the establishment has been extremely far-reaching and has occurred in a very short space of time. So I'm going to just see if I can get this cursor working. Laser pointer, there we go. So from its native Southeast Asian range, it has now expanded to the European continent and as well as the Americas, and it has done this in an extremely short space of time. Although interestingly, it has been found on Hawaii since the 1980s, although it wasn't reported to cause economic impact. But on mainland USA, we found that spread it was first detected here in the state of California. Now, these numbers represent the year of detection, so the darker the color, the later the year. But what we can see is that in the first year of detection, it very rapidly spread in the next year into these northern states of Oregon and Washington. But very remarkably, over 1,000 kilometers to the southeast of the state of Florida within the space of 1 year. And within the space of 6 years, it had established in all but about 8 states in the United States of America. If we look to Europe, what we find is that in the same year, 2008, the first detections were in Spain and Italy. And within 6 years, we're basically seeing it invade the majority of the European continent. So spread occurs extremely rapidly with this pest. But you can see that some of these more cold regions took longer to establish. So how is it able to persist in such a wide range of environmental locations? So one of the key determinants of population growth potential is temperature. And so what we see here is a plot of temperature against population growth rate on the vertical axes. And we see that very little action happens at these lower temperatures, particularly under about 5 degrees. And population growth is highest at around 25 degrees Celsius, but rapidly drops off as we get into these high 20s. So populations don't do very well under high heat regimes, but they are able to persist quite well under cold temperatures. Now, this data comes from constant condition laboratory studies, but in the field, you get extreme cold conditions, extreme heat conditions, and dry conditions as well, which are going to impact on the mortality and population growth of this pest. So what we can do to get a sense of what the establishment potential of this pest is combine this biological knowledge of spotted wing Drosophila with climatic data across the globe to get an estimate of what these establishment potential might be across space. So here we have the mean population growth rate averaged across the whole year. And what we see is that lines up pretty well with the regions in which we see these large permanent populations of spotted wing Drosophila. But we also see regions in which spotted wing Drosophila doesn't occur, but conditions are predicted to be quite suitable. And one of those locations is Australia. So if we zoom into Australia, we see that these eastern coastal regions are particularly suitable, as well as some locations in Western Australia. But as we go further inland, where it gets drier and it gets hotter, these average conditions for spotted wing Drosophila worsen, and so they can't sustain populations. Now that was averaged across the whole year, but if we look at months across the whole year, of course there's going to be variability as temperatures increase and decrease and things get wetter and things get drier. But of course, the population is not going to be able to establish inland. One, because they're only going to be at these favourable conditions for spotted winged Drosophila for short amounts of time. But also importantly, we're just not likely to find an abundance of wild hosts. But where we are able to find an abundance of wild hosts is in these sort of eastern coastal regions. So this here is a plot of the number of records for rubus species, which includes wild blackberry, which is a favourite host of spotted winged Drosophila, which is unfortunately spread extremely widely across Australia, which is likely to improve the spread rates of this particular pest. Which brings us to spread. So what we've done in estimating spread potential is combine this biological knowledge of where it can occur, as well as additional biological knowledge of how fast it spreads. And naturally, it doesn't spread super fast. So some of the best flyers in a particular population were estimated to fly about 9 kilometers in one generation, but we saw that hop between California and Florida occurred in 1 year. So how is it doing that? And the obvious answer is through human-mediated spread. While this fly can't spread very fast by itself, humans are extremely good travelers. And so by incorporating a human-mediated spread module, we can actually predict these jumps that occur from these populated areas such as California on the west coast of the United States to all the way over 1,000 kilometers to Southeast Australia and Florida. And then subsequent spread inland, but hopping across these cities all along the way. Now we can fit that pretty well because we're tweaking the parameters until we get a nice fit. But I think what's really impressive is if we take that fit to the United States and we run it in Europe, we also see an extremely good prediction of how spread unfolded in the European continent. In fact, we see about 82% accuracy in the presence and absences predicted through time for the US. And without any further parameter tweaking, we can still get very high predictability of spread. And I think the reason for that is because of the large role that human populations are playing in the spread of this. And we know a lot about human populations. We can say a lot about where the highest populations are likely to be. And this seems to capture a lot of the spread in spotted winged Drosophila. But all of this is to basically go towards making sure that when we make predictions about how spread is occurring in Australia, that they are sensible. So the good news is that we have pretty good confidence that our spread predictions in Australia are going to be sensible. The bad news is that spread is going to be extremely rapid. So here we're simulating an incursion from Brisbane. Now remember, and this is occurring over 6 years, it's being driven by environmental suitability, so the populations are growing faster where where climate is more suitable. We're seeing hops between cities, and local spread is also occurring. But what we see is over 6 years, spread is extremely rapid across the Australian continent into much of its sort of ecologically suitable locations. So not great news. So we really want to avoid this thing coming into Australia. Which then leads us to impact potential. So just because the pest can spread to a location doesn't necessarily mean it's going to cause impacts. So this is a picture of spotted winged Drosophila. Now it's got the spots on the wings. Now this is the male. What we find in the female is that it lacks the spots. But what it does have is this serrated ovipositor. And this, I think, is actually a defining feature of spotted winged Drosophila because it's this serrated ovipositor, or sharp sort of egg-laying stick that it's got on its bum, that allows it to pierce the skin of soft fruit and actually cause damage in the first place. So what does this damage actually look like in terms of on the fruit? So here we have an incision made on cherry by that sharp ovipositor. And of course that causes impacts directly, and particularly as populations grow, densities increase, and they start causing the fruit to collapse around that incision. But importantly, there's also secondary effects. So there can be mold damage here, so this is secondary infection, as well as secondary impacts from other pests that take advantage of the exposed fruit and start to feed. So here we see secondary damage caused by other beetles that are co-occurring with this fly. So what does this look like in terms of numbers? So now here we review the international literature and other sort of reports on crop losses for spotted wing Drosophila across a range of different commodities. So here I'm showing the top 6 most reported crop losses, and for blackberries, blueberries, raspberries, strawberries, sweet cherries, and table grapes. First thing to notice is that the estimates of proportion crop loss in terms of percentage crop loss are huge. Sometimes we're seeing 100% of crops being written off if the infestation levels are that bad. More generally, the impacts are reported much lower. There's also variation between commodities, where we see that some less impacted commodities like table grapes compared to more impacted commodities like raspberries and blackberries. So how do we explain this variation in impacts? Well, there is just a lot of variation. Some of that's going to be accounted for by different management practices, the location, but I think a really interesting one is when you look at the years from first detection in a particular region. What we find is, although we have, generally speaking, higher impacts caused by in raspberries and cherries, they generally decrease through time. So what does this mean? What are we seeing here? Well, I think this is better the devil you know in this case. So as growers are getting more familiar with this pest, they're improving their management practices, they're improving their crop hygiene, improving their identification, they're getting on the problems early and minimizing their impacts. So really, I think what preparedness is about is trying to get us to start here on the curve. So let's learn from all these cases of high impact overseas and really expedite the process so that we can minimize impacts. But let's just look at worst case scenario. If we have, if these are all the production regions and the value of the commodities produced in these regions, if we overlay that with our spread simulations, we can get impacts for what might impacts be through time if we're not good about managing this pest. So we can run this for a lot of different simulations, a lot of different commodities, but let's just focus here on total impacts of affected commodities. What we see is that, so time since incursion in months, after 3 years, we're seeing impacts estimated of around just under $20 million to over $60 million, depending on where the initial incursion location is. So now, importantly, we've made an assumption here that the impacts are going to be around about 10% across the board, depending on the across all those commodities. And now this was just to isolate the effect of incursion location. So we want to see, is there a particular location that's going to cost more money in terms of it coming in? And we can cut this finer. So here we've just looked at a total impacted commodities here, but we can cut this finer and break it down by the particular fruit and by the particular state. So here we see the impacts estimated for each state, and in this case cherries, as well as different incursion locations that hypothetically could occur in those states. And what we see for cherries is that the highest impacts are estimated for incursions into Hobart, so reaching about $10 million worth of impacts after 3 years, which is a bit worse than an incursion into Devonport, but we're seeing still fairly high impacts in other states, particularly Adelaide and Melbourne, of around $2.5 million estimated impacts after 3 years in Melbourne. And we see that just because that impacts take a little while to accumulate, and that's due to the population spreading and the populations increasing in time. I should also mention that there are these sort of shaded regions around the lines, and they reflect the uncertainty around these impact estimates due to the that spread is a stochastic process. It can happen randomly. Sometimes we'll see a hop between one city and another, and they're very hard to predict. And so these estimates represent that uncertainty. So now if we look at strawberries, we see that this is an extremely high value of the production value of strawberries is extremely high in Australia, largely concentrated around Brisbane, which are expected to see some of the highest impacts due to the rapid spread and proximity to cities in Brisbane, but that there are also very significant predicted impacts across a range of different states, including South Australia, as well as Victoria, WA, basically every state except New South Wales in this case are expected to see large unmitigated impacts if we don't take action against this pest. Caneberries, so your rubus here, raspberries, blackberries, again, a very vulnerable crop and widely distributed across different states, but highest impacts occurring in Melbourne, estimated at around 1.5 million after 3 years. Brisbane also seeing high impacts of $1 million after 1 year. So keeping in mind these are different, these plots represent the different jurisdictions. Blueberries, we see very large estimated impacts for incursions into Sydney and Coffs Harbour, but with large variation around those estimates, reflecting large uncertainty. Whereas incursions into Brisbane and Cairns are expected to be around the order of $2 million after 3 years, with high impacts estimated also for Tasmanian incursions, seeing about $3 million worth of impacts after 3 years, and $1 million in Hobart after 1 year. And this is an interesting situation where we get lower populated regions that are causing higher impacts. So in this case, Devonport, which you can see the concentration of blueberry production in Tasmania is in the northern part of the state. Table grapes is an interesting case because while these inland hot and dry regions where a lot of the table grape production is concentrated is predicted to be largely unsuitable, if there is an incursion, what the predictions are estimating is that impacts could be high initially, but then flatline as that population actually goes extinct at that location. So there could be small impacts that occur, well, not small, these are still in the order of $3 million due to the sheer size of the industry. But due to the extinction or predicted extinction due to unfavorable conditions, due to the heat and the dry of these growing locations, they may be transient populations. Now that was a lot to get through. There's a lot of different scenarios that we can play out. There's a lot of different commodities that are likely to be infected. And so what we've also done is created an online tool where you can explore different incursion scenarios. So in this case, if you go to this website, you click on a point in the map, a spread simulation will be run across a year where the color indicates the likelihood of spread. You can overlay the particular affected commodity value of production by selecting it on the left here, which just gives you a sense of where these impacts could most likely be seen. So if I didn't have time to cover a particular case of interest to you, I encourage you to go online and explore these scenarios for yourself. So in summary, spread has been extremely rapid. There's a large area of Australia that's predicted to be environmentally suitable for this pest, and that spread is going to be facilitated first and foremost by human-mediated means. Incursions into populated areas of the East Coast Australia were predicted to be the most conducive to rapid spread, although across a time horizon of around 6 years, spread is going to be fairly rapid regardless of where it gets into in this case, particularly if it's around highly populated regions of Australia. In terms of impacts, after conducting a literature review, we see that impacts start off fairly high at around 20 to 50% within the first 2 years, but decline with time as management improves, identification processes improve. Australia does have large fruit industries that would be impacted if we're not properly prepared for this pest. and importantly, that these different jurisdictions and industries will see different sensitivities to particular incursion scenarios. But hopefully, through the preparedness outputs that emerge from this project, we can really move further along that curve and reduce these expected impacts. I'll wrap that up because I've gone a little bit over, but I'd like to acknowledge all the project partners, and in particular funding from Hort Innovation. I'll pass it on to Lloyd now, who's going to talk about surveillance.
I'd just like to say, Sharon Taylor, just sort of butting in very briefly. Lloyd will be sharing his screen very shortly. Always takes a little bit of a lag. Excellent, looks like it's coming up. I'll pass over to Lloyd.
Just unmute myself first, there we go, that's better. So thanks for that. Now, as you've seen from James's talk, it's looking like it's going to spread quite quickly if it comes into Australia or New Zealand. So how are we going to look for it? What are we going to use? And yeah, how can we find it? So Dave and I have pulled this talk together, but we've been working with Hort New Zealand, Hort Innovation, of course, and B3, Better Water Biosecurity in New Zealand. Now, just before I go into some of the surveillance tools, I thought to touch on the, there we go, it's moving. I thought we'd touch on some of the work that's already occurred and to get us prepared for this. So both Australia and New Zealand governments, industries and science have been generating information and sharing that information, and that's part of what this is today. You can see that both governments have done pest risk analyses for spotted wing Drosophila. So that's to understand a little bit more about the risks of what we're importing into the country and what sort of mitigations could be put in place to reduce the risk of importing anything. And of course, there's also been a little bit of fact sharing from the industry. So here's a fact sheet taken from the Kiwi Vine Health website, giving a quick overview of what it looks like, some sort of damage it does, and where it can be found. So we've also had the opportunity to speak with people in governments about what has happened in their jurisdictions. So you saw earlier the European and US spreads, and we've had the opportunity to talk with Chileans about the spread of Suzuki in their country. So I'll just acknowledge Ruth and Carlos here for letting me share some of their slides from earlier. So in this local process, so they had traps out since 2012 through the growing areas of Chile, particularly near the south there where you can see the sea. And Chile has its growing area mainly through the middle, so you've got the Andes on the right and then another mountain range just to the left. And they had nothing until 2017, just like that. And you can see all the dots on the screen there. They had a big, a large number of traps triggered almost simultaneously. The next year, you see that spread has gone quite a way up the country. And those maps are about 600 k's length each. That's a very rapid spread. And for those who like graphs and figures, so you can see what happened in terms of the trapping here. So this figure here, we have time along the bottom axis there. So May 2017 through till September, and the number of Drosophila Suzuki trapped. Initially very low numbers, that following year those very high peaks. So what that equates to in catch ratio is in that first year, there was for every one spotted wing Drosophila detected in a trap, there were 600 other Drosophila species. And they have the added excitement of having another species that looks like male spotted wing Drosophila with the two spots. So it makes diagnostics quite tricky for them. That following year, that ratio was 1 to 1. Still same numbers of flies being caught in the traps. And there's the picture of the trap they use there at the bottom left, but I'll hold on for that for a moment because I'll give you some more detail in a bit. So we know from previous work where we've analysed eradication and incursion responses from around the world from this database, which you're freely welcome to access and get some of the data, that if you can detect the species early, you have a higher chance of eradicating it or at least having better management options. And that is facilitated a lot, so you get a 20 times increase in chances of eradication at least, by having a lure. So you can actively sample for the species, or a species. So what sort of detection do we, or detection system do we have running in New Zealand? So on top of the import health standards and everything that conveys the sort of processes that commodities need to go through before they're allowed into the country, there are still checks at the border. And some of you may have seen in the news, there was a detection in an orange from a consignment of oranges from the US at the border. So before being allowed into the country, 2019. And before this, there was Drosophila Suzuki detected from Asian origin in 2012. And New Zealand has a general surveillance system as well running. So this is a free phone number that anyone can call, so industry, public, government, when they see something a little bit weird out in the environment. And from there, that risk can then be assessed, and there may be someone who comes out and does some follow-up. And then for Suzuki and specifically, there isn't anything active at the moment trying to detect it as it arrives, post-border that is, but there are a number of lures that have been developed and have been tested. But just before I go on to sampling for the adults, you can sample for the larvae. So our colleagues at EMR in the UK have a great video here, it's the link below, where you can sample for larvae in fruit. So instead of having to go through and troll through the fruit manually, making a heck of a mess and probably missing a lot of larvae, what they use is a flotation technique. So they have a saturated sugar solution in which you add the fruit, slightly broken up, and this encourages the larvae to escape, and then they float to the top, making it much easier to actually sample the fruit and see if you have larvae in the fruit. Now, in terms of sampling for adults, initially apple cider vinegar was used, and it is still used. It's really cheap. A lot of you will probably have it in your cupboard. So it's really available, but it isn't particularly specific. So you can see in this figure here, we have some light brown apple moths, some ants, some cockroaches by the look of it. And they've all been sampled by this apple leaf curling image, by this apple cider vinegar. And with this apple cider vinegar, the liquid needs to be replaced reasonably frequently. One from evaporation, but also you just get so many insects in there, and all the liquid sort of disappears, and you've got them brimming over the top of that trap. Now, another non-synthetic lure and trap combo is this Biosecurity lure. So again, it's just mainly fermenting odors from vinegar, red wine, and some sugar in there. And it's pretty unlikely to have regulatory issues because you can buy it off the shelf. Well, you can't buy it off the shelf yet, but there are stockists suppliers based in both New Zealand and Australia for Biosecurity. The lure replacement is fortnightly, and this combination of the colour and the odour greatly improves trap catch. Now you'll see on the picture of the trap there is some little round circles, and those are like a filter system they've put over quite a large entrance. So again, it's still bringing in a large number of various species beyond just Drosophila, so they've tried to filter it physically just by making it a smaller hole for the insect to get in. Now, so initially there were a lot of these raw lures, stuff you could buy off the shelf, but now quite a few groups have been trying to develop longer lasting synthetic lures. So both to get that additional time in the field, but also to reduce the bycatch, because that is a massive issue for anyone looking through those traps, especially when they're wet like that. So it's recommended that these lures are used with a wet trap, so some sort of water solution to catch the insect, drown it, and retain that sample. But I can't see any reason why a dry trap could not be an option if we had the right sort of killing agent in the trap, which may help you make friends a bit more with the diagnosticians who need to look through those samples. Now, the lure replacement for these is about 4 to 6 weeks. However, there may be some issues importing and using these, as they do contain a number of chemicals, which can be quite hard to get for proprietary reasons from the companies. So it makes those conversations with the regulatory agencies quite interesting. So you'll see the samples here. So there's Alpha Sense does one, Sentry, which we'll go into a bit more because they've published quite a bit of information. And Trace has a couple of lure combinations. So one is this red one, the high specificity one. So that reduces the bycatch of other Drosophila. However, you also have a reduced trap catch, and then they have a broad spectrum, which is that massive lure. So pumping out a whole lot of odors, but bringing in a vast variety of insects. So, James has touched on this. I'll just show you a couple of other figures, but the diagnostics, some of the black wing, the spots on the wings for the males, it's mostly always there, but sometimes it isn't. And then, of course, they've got the sclerotized ovipositor. But on top of that, there are people looking at molecular tools to diagnose what you have in the trap. So you think of these two lures again, that's a pretty ugly sample to go through. But there have been some primers developed that will help you sample the insect and be able to tell if you have Drosophila Suzuki or not. So at the moment, it's at the point where you can do insect at a time, but there are groups looking for improving the methodology so that large numbers of samples can be processed at the same time, and also to get that response, or the is it one, yes, no, quicker. Now you can, one of the bonuses with this sort of diagnostic is you can just sample the liquid, so you don't need to homogenize or mush up everything. So you can actually go back to the sample later on if you have a positive detection and confirm that with the actual sample. So there are some lures out there, but how many should you put out? How far apart? And what's your chances of catching anything? So as I mentioned earlier, I had some good fortune of Sentry, or a PhD student, publishing quite a lot of information from using the Sentry product. So she used the Sentry lure with a red panel trap, so a sticky panel trap, and did a whole lot of mark release recapture trials. So marking all these insects, releasing them at different distances from a trap. And as you can expect, the closer an insect is to a trap, the more likely you are to trap it. And as you move further and further away, that insect is less likely to be attracted and trapped. That's quite a difficult shape to model. So using some earlier publications, we can take that curve, or that curve, and change it and push it into a cylinder of a unit height to give us a correlation coefficient, or the ESA, the effective sampling area. And from there, we can start predicting how likely an insect is to be attracted and then trapped by a lure. So here we've estimated what that probability of detecting a population of different densities is with different densities of traps. So along the x-axis here, we have the population density of trappable insects, so 100, 200, 300 insects in a hectare, and that daily probability, so the probability of catching any insect in that one day, and a different number of traps in those different colours. So 100 traps per hectare is green, and of course you'd expect if you have more traps in the area, you're more likely to trap the insect, and as you have fewer traps per hectare, that probability of detecting any of the flies in that population reduces quite significantly. So that's, that was trapping for one day, but of course we tend to have traps out for a lot longer than that. So what is that probability of trapping spotted wing over time? So in this case, we have a population of just 10 trappable individuals, assuming they've stayed alive for this whole time. And we see that, again, 100 traps, very high probability of detecting one of the flies in a day, in a few days, but also 50 traps. Also, we do not have to sample the area too long before we have a high likelihood of detecting an insect. Of course, once you get down to 10 traps or 1 trap per hectare, that probability of catching a fly is much reduced. And of course, the longer you take to catch it, the more time that fly or population has time to increase in size and density. Now, as well as some of that earlier work we showed you, we're also linking with other groups globally, trying to work with them to understand some of the new tools that are being developed. And here's one of the examples. There's a collaboration with a number of countries, 5-year programs, where we're looking at new biocontrol agents for managing spotted wing, the sterile insect technique. So anyone who works with different flies knows that drosophila can be a bit of a pain in the rearing factories, and this is quite rearable, and there have been quite a few papers come out of late in terms of rearing techniques and the irradiation biology. And of course, it's how to use those. So do we want to apply all the tools at once, simultaneously, different times a year, and the like? And here we've got a picture of some drosophila there on the left, just in a little cage. And then one of the trials in Argentina where both biocontrol agents and sterile insects have been released into a population, and we're looking at the number of larvae or eggs that have been laid in both strawberries and a matrix here, just a overposition matrix. So some of the take-homes from today. So there are a number of lures out there, and they vary a lot in their specificity and their availability. And while some of the newer lures are more specific and targeted and longer lasting, there may be a few approvals to get first prior to being able to import and use them in the field. On top of that, there's some new diagnostics for mass diagnostics being developed by groups. As far as I know, nothing's ready yet, as well as new tools. But really, the longer we can keep SWD out of both Australia and New Zealand, the better equipped we're going to be should we finally get it, and of course reduce that disruption to industry in the long term. Thank you.
All right, it's Sharon Taylor back again. I am just going to share my screen back. Apologies, this is taking a little while. Great. Excellent. And thank you, Lloyd, for that picture of surveillance and the fact that while we have some tools available, it is a very difficult pest to actually look for. Now I'm going to share some information that we've got on management and control options for spotted wing. I would preface this by saying I'm not the expert in this space, but have certainly been part of the team that's pulled together this information. What we're going to look at today is control options, and really just reinforcing the point that integrated pest management is going to be the key for spotted wing Drosophila. There is no single method that controls this pest, and it certainly has caused challenges overseas. So I want to reinforce that part of the integrated pest management approach really does rely on monitoring. Lloyd's gone into all of the different techniques that have currently been used, and it's taken overseas partners a long time to actually get some of these techniques sorted. But certainly when we're looking at control options, monitoring, as with a lot of pests, many pests, it's really vital. It allows management decisions to be made and is a really strong part of a decision support system. And we're starting now to get some lures which are more specific. In terms of chemical control, I'm just going to give a high-level overview here. There are chemicals that are available that can kill adult flies. Probably not surprisingly, there's a suite of actives that are relied on overseas. The usual suspects of the pyrethroids, organophosphates, carbamates. A lot of these are broad spectrum. That is, we know that they're going to knock our beneficials around. In doing this, and certainly that's what we see when many new pests enter our production systems, and they've certainly done this overseas, it causes not just issues for managing the pest itself, but then causes spikes in secondary pests that come through once IPM has been disrupted. There are promising technologies coming forward with attract and kill, or what are called splat technologies, which are specialized pheromone lure application technologies. None of those appear to be currently registered from what we can see, but certainly the work is progressing, and some of those attract and kill where you actually have a lure which brings the adults in and then actually has a chemical to kill them, certainly showing promise. I'm not going to run through them, but this project's identified a range of actives which are effective in overseas use. We've also compared those to current use patterns in Australia on different crops. So we've been able to see where with not too much effort, fingers crossed, we should be able to get emergency permits in place for actives that are currently in use, but we will need emergency permits if the pest arrives. But we've also importantly found gaps where we do not have actives in certain crops that we're going to need, or at the rates that we're going to need them. And so it's highlighted where data will be required, either for emergency permits, but particularly if we want to get stuff on label. Cultural control is where I'm going to spend a bit more time because there's a range of different things that have proved to be highly effective in a systems approach. I've got them listed here, but I'm actually going to run through them in more detail now. So one key tool in the arsenal is microclimate manipulation or canopy manipulation, really trying to reduce the microhabitats that spotted wing Drosophila likes. And that can be done by opening and thinning canopies of different crops by pruning. Also managing the irrigation to make sure, and in Australia sometimes that's difficult with our low water supplies, but managing irrigation to limit fruit splitting and reducing humidity. If we reduce fruit splitting, we reduce harbours for or easy spots where adults can come in and actually lay eggs. Also anything that seems to increase airflow and reduce shading, so canopy and wider spacing, or thinning canopy and wider spacing. The flow-on effects of all of that are that there's less chance of fruit being missed in harvest. And that brings me on to harvest frequency, because harvesting often and early can disrupt the spotted wing Drosophila life cycle, and particularly removing fruit as soon as it ripens, really shown to be really important in certain crops. in soft fruit, like the caneberries and cherries, harvesting, or caneberries I should say, and strawberries particularly, harvesting soft fruit every 2 to 3 days has shown to be very efficacious in keeping numbers down. Moving on to another really important tool, hygiene and sanitation, and this is probably where people have had great success, but it's also comes with cost, because getting in and picking early and often, but then removing all ripe fruit or all damaged fruit is very important, making sure there's no fruit on the ground. And a simple way of actually destroying fruit has been found, just putting it in bags, sealing it up tight so the adults can't escape, and then letting it cook in the sun. We would have to do similar research here at overseas situations. They say that fruit needs to be left in bags sealed tightly for more than 32 hours. So we'd have to see if the same happened here if we're having slightly higher temperatures. And obviously removal of woody weeds, so removing reservoirs and areas where the adults can breed. Other parts of cultural control, cultivar selection takes time, but has been part of a systems approach. So looking at early ripening varieties, and also if there's a potential for thicker skinned varieties. Apologies, just had to take a sip of water. The use of exclusion netting is costly, and certainly we've heard from overseas that prior to spotted wing arriving, growers would never have considered exclusion netting, insect-proof exclusion netting as an option. But they're considering it now. It's only as good as the netting itself, and it's only as good as keeping movement in and out of structures as low as possible. So it comes with issues. Weed matting has shown some promise. Fabric or plastic, significantly better than no matting at all, or use of mulching like wood chips, and that's because it's limiting the pupae in the soil. And also the use of mass trapping, that's certainly still being investigated. So mass trapping, planting susceptible crops near crops that are less susceptible. Raspberries are extremely susceptible. So planting sacrificial crops and using them to draw the adults in and then destroying. Certainly trapping itself is very important for monitoring for spotted wing Drosophila levels, and therefore knowing what sort of controls to put in place. Biological controls, I'll just quickly touch on. Biological controls are being investigated in a number of countries. Suspects such as fungi, bacteria, viruses as biological control. Importantly, just beneficials, natural enemies. And what we've got is a project underway in Australia, which Cesar is part of, a component of which they're actually looking at reviewing Australia's natural enemy options. It looks like soil-dwelling natural enemies will be our best source of biological control in the first instance, so things like earwigs and carabid beetles. As Lloyd mentioned, sterile insect technique, certainly it's used for fruit flies, and it's being investigated for spotted wing, and incompatible insect technique. Both of those are just techniques which sterilise males, and then males can be released, and that will disrupt life cycles. That was a very, very quick run through of take-home messages. A lot of the extension material that's been produced will also help with some of those messages and provide more information on some of those messages. So take home, really there's no magic bullet. There's no single chemical or no single approach which manages spotted wing. It is going to be a systems approach, and it is going to take changes to current management systems. Aspects of cultural management will be key. Monitoring will be very important, but improving hygiene overall seems to be a very important take-home message, and canopy management has been useful overseas. I think what we can see from all of the work that we've heard described, James's work looking at spread, Lloyd's work with surveillance, has shown for us in Australia and New Zealand, eradication is going to be extremely difficult because it's going to be a hard pest to find, and it's going to be a hard pest to find early enough. Growers in the UK and Europe certainly saw the potential for spotted wing arriving and could see it arriving on their doorstep. And as it arrived, what they saw as beneficial, or what they got benefits from, were awareness. So things that we're doing, like we're doing now with this webinar, improving awareness about spotted wing Drosophila, what it is, how we monitor for it, how we can put monitoring in place. And then starting to look at systems to improve hygiene and waste fruit disposal prior to its arrival will actually help everyone transition should spotted wing arrive. We very much hope it doesn't, but it is a cryptic pest and it's going to be a difficult one to keep out. That sort of ends the main part of the talks that we've had. We've discussed a lot about, we've had talks today on spread and dispersal and impact, surveillance. We've touched on management and what we know about management. This project, and as I said, Jessica Lai from Cesar has been very instrumental in pulling a lot of this information together. There's educational packs or resource packs that we've been producing. We've got videos that have been produced. We've now got 2 webinars which will be available. James has produced an interactive prediction map, and we've got other articles and information. We're also, to help our decision makers and policy makers from both industry and government in both countries, we're putting together a preparedness plan, which is actually a compilation of all of the information that we're bringing together in all of these areas. And this will help us actually respond to spotted wing should it arrive, and certainly helps work out where our gaps are in the information that we currently have. This project itself, that you've heard all of this information from, is due to end in June 2020. There is still one Hort Innovation project which is continuing. So there still will be work going on in preparedness in this area, and we'll be bringing together recommendations on where gaps still appear. There's a couple of email addresses here, so please feel free to contact us if you want more information on this project or on what we know of research going on. And as Jess has said, hopefully people have been watching the comments and replies coming through. We will be able to release this webinar hopefully next week. We'll be bringing everything together. What we'd like to do is actually rerun the poll that we asked before. Probably brave of us, but we'd like you to put in your responses to this. Please be honest in terms of whether this has helped increase knowledge, but certainly too there's other areas. If you need more information in other areas, please get in touch with us, and I'm sure we've got information, or we can point you in the right direction. So I'm hoping if I move to, while that's going on, this is just a reminder to everyone. If you think you've seen spotted wing, it is a very tiny, small fly, smaller than Q fly, that we'd be more familiar with, the Queensland flies, well, for those of us in Australia. If you suspect you've seen spotted wing Drosophila, please take a photograph, or particularly collect a sample of the pest or the fruit. Definitely take the location. Call, there's a couple of hotlines here. Please get the right one. If you're in Australia, call the Australian one. If you're in New Zealand, call the New Zealand one. Call before you send anything in. And we've just got the results of the poll. So we've done well in terms of the webinar today. We had most people feeling that they had a low knowledge of spotted wing Drosophila. We're now up to moderate, which is great. And so what I'd like to do, we're actually just on the end of the time that we've allotted, and I know it is later in New Zealand. As I said, we've got a couple of contact points there. We will be circulating the answers to some of these questions. I hope we've managed to get onto questions, and I hope while I was talking, people have managed to get onto questions if they've had anything on management. but we will be circulating that, and as we said, we'll be circulating this webinar with this information hopefully later next week. So with that, I'd really like to thank the project team, and I'd love to thank everybody for actually participating. It's a shame we had to do this as a webinar rather than face-to-face, which was our original plan, but it does mean we've actually managed to have more attendees than we probably would have managed as a face-to-face. So with that, unless any one of my panelists here has anything further to add, I think we'll be calling it closed, and thank everyone for their time.